Welcome to lesson 6.4. In this lesson, we'll talk about the role of protists in soil nutrient cycling. We'll specifically look at how the protists that feed on bacteria, called bacterivores, how these protists affect nutrient cycling in the soil, how the protists that feed on bacteria modify the microbial community within the soil and affect the microbial populations. So let's jump into lesson 6.4. Soil protists are extremely biodiverse in soils. A gram of soil can actually contain thousands of species of different protists. So if we were to look at a nail-sized amount of soil, that would be about a gram, the biodiversity in that is equivalent to that of the insect diversity per hectare in a tropical rainforest. And yet, soil protists are the most understudied group of organisms, and they are often not even considered in ecological or biodiversity studies of soils. So many scientists are now really speaking up about the fact that soil protists need to be considered in soil studies. There is no point in looking at soils without actually acknowledging and studying the biodiversity as well as the ecological role and function of soil protists. And this is because they play an extremely important role in nutrient cycling, and they have so many various ways of feeding. They're just so diverse when it comes to their feeding behavior. So some soil protists are phototrophs, so autophototrophic, that means self-feeding via photosynthesis, via carbon fixation. And they are particularly important in desert soils where they play a role in the formation of cryptogamic crusts. And this is a collaboration between protists, fungi, algae, cyanobacteria, and bacteria in creating these crusts that hold soils together. And we'll have a slide about that later. Osmotrophs absorb dissolved organic matter via the cell membrane, and oomycete group of protists is really good at that. They actually just absorb their nutrients from the surrounding environment. Phagotrophs engulf other organisms, so they're predatory. They take in bacteria and other protozoa, smaller protozoa, even fungi, and organic matter, of course, as well. So this is by the process of phagocytosis, when a cell engulfs a particle or another organism. Interestingly, some osmotrophs are phagotrophic, so that means that they can use two modalities of feeding. They can absorb nutrients via their cell membrane, and they are also capable of engulfing either organic matter or being predatory. And we can either call them phagotrophic osmotrophs or osmotrophic phagotrophs. Just depends how you want to phrase it. There are also mixotrophs, and these are phagotrophs, so they feed on organic matter or engulf other organisms, but they are also photosynthetic. They have either chloroplasts or have symbiotic algae living within them, allowing them to photosynthesize. So all of these are heterotrophic, and the only autotrophic, i.e. self-feeding or making their own food, are the phototrophic protists that photosynthesize. But the rest are heterotrophic, so they need carbon from other sources. They can't make their own carbon like phototrophs do. Unfortunately, most heterotrophic protists, particularly in soils, are almost always treated as bacterivores. The notion is that most of them just feed on bacteria, and this is very untrue because some of them specifically feed on algae, some specifically on fungi, so they either algivorous or mycophagous, and some hunt nematodes, and these are the nematophagous protists. Some are strictly parasitic, and some eat other protists. So they are so diverse in their dietary requirements or their dietary preferences that it is very, very important that these organisms are acknowledged in biodiversity studies and ecological studies because they play such a huge role. They have so many interactions in the soil food web with their prey or their food source. And this is just a beautiful image of all the different types of soil and aquatic protists as well. That's an alga actually that contains protists inside. It's got flagellates inside. And another creature that I wasn't even aware of is this amoeba that is, unlike these three, which are testate amoebae, this one is only half-shelled or half-naked amoeba. So up the top it's got a shell like a snail, and on the bottom it's naked, and that's a fully naked amoeba. So that's just really, really cool. And I highly recommend reading this relatively short article called Soil Protist Life Matters. A few soil protist researchers got together and wrote this really quite concise and lovely article about the importance of soil protists. I highly recommend reading that for yourself. So let's look at the heterotrophic soil protists that live via predation or grazing on other organisms, and specifically let's look at their relationship to bacteria, feeding on bacteria, and the effects that has on the soil ecosystem. So phagotrophs feed either on organic matter or ingest microbes like bacteria or other protists. And one study has found that 258 species of soil protists from the rhizosphere included 63 flagellates, 98 amoebae, and 97 ciliates. And 40% of those were found to be strict bacterial feeders, 36% were mostly bacterial feeders, 20% ate other protozoa, some ate fungi, some detritus, and some consumed algae. Different species of protozoa prefer different bacterial species, and some actually might be very related, but will have very different dietary preferences when it comes to their consumption of bacterial species. On the other hand, some bacteria are very prone to be eaten by different species of protozoa. So in one study, 3 different protozoa fed different types of bacteria, all preferred the one bacterium species. So it just really depends on the types of bacteria and really depends on the food preferences of the predators or grazers. Also, some bacteria are actually grazing resistant, making them inedible to various protists. They may produce toxic compounds, various pigments that the protists don't like, are hydrophobic, so that means water repellent, and they can also change their cell size to avoid being predated on by growing larger cells, switching from bacteria that were once a certain size, but now the new generation is increasing their size in order to avoid predation. There are also some of them that start forming colonies or already are colony-forming bacteria to prevent being wiped out by protists. And also some are motile, so they can actually escape. They can be chased, of course, but they are more likely to escape if they are motile and can move away from the grazers or the predators. That's just some of the mechanisms with which bacteria protect themselves from being eaten by soil protists or protozoa. I really love this image. It actually is an amoeba, and it is hunting, and it has engulfed, and it's consuming diatoms. So these are all diatoms, and it's consumed a diatom, and it's holding it in a digestive vacuole. So that's a really cool image of how some of these predatory phagocytic or phagotrophic protists, such as amoebae, feed. So those phagotrophic protists that feed on bacteria are called bacterivores, and they influence bacterial populations through their predation. And this is really important in nutrient cycling because the protist has a greater carbon to nitrogen ratio compared to a bacterium. So that means every time a protist consumes a bacterium or bacteria, it actually releases ammonia and other nutrients, which become immediately available to the plant around the rhizosphere. The plant will only consume a certain amount of these nutrients, of this excess nitrogen and other nutrients, so the rest will actually go directly to feed other microorganisms, such as osmotrophic protists, which actually absorb the nutrients directly through their cell membrane. So basically, the moment a bacterium is consumed, and then through the digestive process of the protist, the excess nutrients are then released. That stimulates plant growth and biodiversity in the soil, as there's food now available to all the organisms present in the soil. Most soils will have an enormous dormant protist seed bank, where there's potentially 10 to the power of 4 to 10 to the power of 7 protists per gram of soil. And these dormant protists can become active in suitable conditions, and that usually happens when there is an increase in bacterial prey. And some of them have extremely rapid cell division, so they can actually double in hours. So they can really cause a lot of changes in the soil ecosystem and really diversify that ecosystem. As these organisms become very prolific, it really influences the bacterial population and population of other organisms as nutrients become available. So let's examine the specifics of nutrient cycling and how it actually looks in terms of numbers when protozoa or soil protists actually hunt or graze or predate on other soil microorganisms such as bacteria. So let's look at the case of a flagellate that has 30:1 carbon to nitrogen ratio, which consumes bacteria, one of which has a ratio of carbon to nitrogen of 5:1. So in order to fulfill its carbon requirements, the flagellate needs to consume 25 extra carbons because it's only getting 5 from one bacterium but needs 30. So it needs to consume 6 bacteria to get enough carbon. And now, as a result, it has excess nitrogen and also other nutrients that the bacteria is richer in than the actual flagellate is. So it has to release 5 nitrogens in the form of ammonium ion as excess nutrient. So it releases 5 nitrogen atoms for every 6 bacteria that it consumes. And let's say it eats on average 10,000 bacteria per day, and that's how many bacteria these organisms actually consume. So that is equivalent to 8,000 ammonium ions released per day. And in a gram of healthy soil, there's about 50,000 protozoa. And these then eat 500 million bacteria per gram of soil per day. And that's equivalent to 400 million nitrogen atoms, which is equivalent to 7 nanograms of nitrogen per centimeter square of root surface per day. And the most rapidly growing plant on Earth, called Arabidopsis, requires only 0.2 nanograms of nitrogen per centimeter square of root per day. So that means that 5.8 nanograms of nitrogen are left in the soil, and that's for fungal and bacterial consumption, and other organisms as well, like the osmotrophic protozoa, for example. So this is how nutrient cycling works. When an organism consumes a richer source of nutrients, such as bacteria, and then has to release those in a plant available or bioavailable form for plants and other organisms to consume in the soil. And this is why it is so essential to have these bacterial predators in the soil in order for nutrient cycling to work. If you just have bacteria in the soil around your plants, which is often the case, and there are no predators, your plant cannot be fed. And this is where you start having to fertilize the plant. But naturally, these bacterial predators will actually do the job. That's their role, is to feed the plant via this system. It's a beautiful, beautiful system. And in the image on the right, that is not a flagellate, that's actually an amoeba. And these are yeast cells, so it's hunting yeast cells and releasing those nutrients from those yeasts in a similar way that this flagellate has done with these bacteria. And these are calculations based on real numbers. So how does the predation of soil protozoa affect microbe diversity? Well, there's selective grazing, which can reduce or eliminate specific species. For example, if one species has greater densities then the threshold predation rate, it actually will dominate the microbial population, versus one that grows slower than the predation rate, which will lead to its elimination. So fast growth rates of microorganisms lead to predation survival versus slow growth rates, which means that the bacterium may not be consumed, but might be expelled. So these bacteria actually prevent themselves from being degraded by the protozoa. Fast bacterial growth rate means that there's continuous replacement of populations of that particular bacteria above the grazing rate. And then there are also those bacteria that have a slow rate of growth, but increase in size, and so they are then too big to consume. And some bacteria actually adhere to the surface of the protozoa or the protist and actually get taken to new environments to populate new environments. And obviously, this is all dependent on abiotic fluctuations, so like moisture availability and temperature of the soil and any pollutants that might be present. But these are the kind of relationships that the grazers or the predators have with their prey, and it influences that prey-predator dynamics as to the availability of the different organisms that it can predate on and their survival mechanisms or their resistance to being consumed or to avoid consumption. Predatory protists also influence microbial community and population dynamics. Through nutrient mineralization, they actually influence the microbial community size, activity, and composition. So depending on the types of nutrients and depending on the amount of nutrients that they provide via consumption of microbes, they will actually influence that community because they will feed the live microbes that are remaining, and depending on that community and how quickly it can feed on those nutrients and how quickly it competes for those nutrients, it will actually be influenced through that predation process. This leads to interspecific competition via nutrient availability. So that's about resource diversity. So that depends on the diversity of the actual grazers or the hunters or the predators in the first place. And then this influences microbial spatial heterogeneity, the variability in the microbial community across space. So how the different microbes are distributed in that microbial population. Grazing pressure increases nutrients, microbial activity, and growth. So when there's pressure on the bacteria, when they're being consumed and their nutrients are being released, that stimulates other microbial activity and growth, and that means also instant renewal of the bacterial or other microbe community. And the fact that a population of microbes is being grazed, that also causes them to want to replicate more and therefore renews the population of that particular species if it's not being consumed at a fast enough rate. Versus elimination. If they don't grow fast enough after being grazed out, if they don't reproduce fast enough above that rate of consumption by the protozoa, by the protists, then it might eliminate that entire species out of that particular spot of soil. So this is the effect that selective grazing and grazing pressure actually have on microbial communities and population dynamics. Selective grazing leads to microbial community composition, structure, diversity, its functional stability, and it increases microorganism turnover. So just a constant change is occurring through that selective grazing pressure. Also, some protists release organic growth factors, which stimulate bacterial activity so they can hunt more. So it's basically tricking the bacteria to divide more, to become more active, so that the protists can actually graze on that bacteria. And those organic growth factors change the microbial community and population dynamics as a result. So very important organisms because they control the soil food web so much via the microbial community and population dynamics that they create through their various grazing strategies. And the greater the diversity of protists within the soil, the greater the complexity within that ecosystem in terms of nutrient cycling and in terms of the population dynamics and community structure of all the microbes within that microbial community. So this is the end of lesson 6.4. And in our next lesson, we'll continue looking at soil protists in nutrient cycling and look at some other food preferences that various protist groups have and how that impacts the soil ecosystem as well as the nutrient cycling within the soil. So you'll be hearing from me again in lesson 6.5. See you then.